Today I'm going to discuss about vaccines and immunotherapy, and this is the last lecture of module 10. In this lecture, I will be mostly covering about vaccines and how vaccines work, and particularly different types of immunotherapeutic strategies, mostly like key concept and the challenges. So here, like first I would like to introduce vaccines, particularly the difference between vaccine and drug. So if you see, whatever drugs, medicine generally we take, so those are used to treat or manage some kind of diseases. But the disease has already happened, right? So to take care of that particular disease, we take drugs. But on the other hand, vaccines are preventive. And more importantly, that drugs, they usually work directly by targeting some kind of pathogen. It can be bacteria, it can be virus, or they can target some kind of body's processes to treat or manage diseases. So if I give you some example, like antibiotics, for example, tetracycline, streptomycin, doxycycline. So those antibiotics are used against bacterial infection. Similarly, like different types of pain relievers can be used to reduce pain. So all these are examples of drugs. On the other hand, vaccines, so vaccines, they train our immune system to face various existing disease causing agents. So sometime I discuss about huge success of vaccination against smallpox virus, right? So in this case, what happened in case of vaccination, it generates memory against specific pathogen, so that if the same pathogen again infect us, we will not have the disease, we will not show the symptoms, because already our immune system knows how to tackle that particular type of pathogen. So this is vaccine. So now I will go into how this vaccine work. So before that, let me explain in case of natural infection what happens. So in case of natural infection, like first time when we are exposed to some kind of pathogen, some kind of virus or bacteria, so what will happen? Our immune system will try to protect us, but that response, the immune response, will be slow, and pathogen will multiply. Finally, we will get some kind of symptoms, and that we said this is the disease, some kind of deviation from the normal physiology. And how much symptoms will be there, how much disease will be there, that is depending on the pathogen itself, right? And also our host physiology. But the thing is, everything is slow during the first infection, and finally after some suffering, after some symptoms, we will have the memory. Memory means here immunological memory. And again, if the same pathogen infect us, I would say 6 months later, sometime it could be 6 years later, but during that time, our immune response will be very, very fast. Pathogens will be disposed soon, and there will be no symptoms or no disease. That's why we will not be able to understand that we are infected by that pathogen. So this is how, our immune system always try to protect us from that same infection. But the same thing can be happening here by vaccination. But in case of vaccination, we use some kind of weak or attenuated strain of that particular pathogen. It can be very weak pathogen, or it can be some parts of the pathogen. So as a result of that, it will not multiply inside the host. So there will be no disease. But whatever we are administering in host body, it will teach the immune system to make memory. So as a result of that, immunological memory will be there. And in future, when that particular person will be naturally infected by the same pathogen, during that time, our immune system will work very fast, pathogen will be disposed and no disease, right? Just like whatever I discussed in the top panel. But here, what is happening, that means when we are like, providing vaccine, then the disease is not appearing at all. So as a result of that, I would like to summarize that vaccination protects us from infection by inducing the adaptive immune response, but bypassing the need for a natural infection. So in the top panel, I mentioned that because of the natural infection, our immune system, they learn how to tackle this pathogen, and they kept some memory, particularly memory B cell, right? But in case of vaccination, all those things are happening, but we are not getting the disease. Now, vaccination, I would say it's a huge success if you think in that way. So it has yielded some of the most successful stories in taking care of worldwide mortality rates. As I already mentioned, in case of smallpox vaccination, it has already been eradicated. And similarly, if you see vaccinations against polio throughout the world, particularly I would say also in India, it is a huge success. Now there is no polio. So polio vaccination is also a huge success. So as I just mentioned, like smallpox, the natural occurrence like last reported in 1977. So it has completely been eradicated. So now, so as a result of that, what I would like to tell you, during the last module, when I was discussing about microbes, during that time I mentioned that antibiotics, those are the kind of greatest invention in terms of medicine, how they protect us from different types of pathogen. Similarly, vaccination is another one. So I would say antibiotics and vaccines, they are kind of major pillar in biomedical science, and kind of we get a lot of benefit out of it. Now, what is the consequence of eradication of that particular pathogen? I would say then the universal vaccination not required, because it has been already eradicated. So there's like no requirement of vaccination at all. For example, for smallpox, now no vaccination is required. And then this is the last point of this slide, the herd immunity. What is herd immunity? So the idea here is, if in a population, if majority of the member, I would say, if they are protected from a particular pathogen, then it is very unlikely that other people will also get that infectious disease, because like that infectious agent cannot be spread, because already most of the people are protected from that particular pathogen. So this is called herd immunity. That means if a critical mass of people acquire protective immunity, they can serve as buffer for the rest. So as a result of that, most of the time they will not get the disease. So this is called herd immunity. So here you don't need to go through the details of this slide. I'm just trying to show you that in different countries they have different types of recommendation for immunization. This is particularly, I would say, child to adolescent stage, 0 to 18 years, and this is I would say like recommended by Indian Academy of Pediatrics. So they have recommended this kind of immunization schedule. And if you see, like some of them, like I would like to mention, for example, in India, BCG is must. So BCG they provided at the very beginning, during like just after the birth. And then you can see hepatitis vaccine, and then DTaP. This is also very common, diphtheria, tetanus, and pertussis. And here rotavirus, for example. So then measles virus, here mumps, measles, and rubella. So this is some kind of a vaccine against some virus. And so as a result of that, we have some kind of recommendation in our country, and all kids, like child after birth, they are taking all those vaccination in a routine basis. So this is, I would say, great benefit for their health also. Similarly, I'm just showing here in United States here what is their recommendation based on the CDC. So they have also their own recommendation. But if you see, most of them are very similar, like almost kind of overlapping with each other, like what kind of vaccines they are also recommending. So, for example, here the BCG is not mandatory. Whatever I told in the previous slide in context to India. So, but here if you see that Hep B, that hepatitis B virus, like vaccine against that, and then rotavirus and DTaP, like diphtheria, tetanus, and pertussis, those are common, same thing. Similarly, in this list, I'm sure that you will find mumps, measles, rubella, that should also be there here, measles, like MMR vaccine. So, as a result of that, now we have very robust, schedule for immunization, particularly for kids. Now, I will start discussing about immunotherapy. This is one of the important, aspects of biomedical science is coming up, and a lot of research is going on like immunotherapy. So then, what is immunotherapy? So this is, I would say, a treatment strategy that uses body's immune system, right? So I would say in case of vaccination also kind of similar. They are also trying to generate some kind of memory in our immune system, right? But the difference is vaccine is preventive, but here because of the disease, we are trying to manage that disease by this kind of immunotherapeutic approach. So as a result of that, a treatment strategy that uses body's immune system to fight diseases, and it can be different types of diseases, but especially against cancer. And it works by either stimulating the immune system to work harder, but it can also dampening the immune response. Sometimes it is also required. This is not always true that it stimulates the immune system to work harder. It's kind of what kind of immunotherapeutic reagent we are talking about and what kind of disease we are managing. Depending on that, we have to think, right? I will explain both of those things. And either by this way or by providing some key components such as antibodies or some proteins that help our immune system to identify and attack abnormal cells. So therefore, we can try to manage that particular type of disease. Now, there are different approach and strategy. I will mostly discuss about two important strategy, and since I discussed about antibody during the last lecture, so it will be very easy for me also to explain this. So one important thing is monoclonal antibodies and engineered antibodies. So what is monoclonal antibodies? See, for therapeutic purpose, generally, like when we try to raise antibody, we try to get antibody, how we will do that? Generally, we use some kind of animal model. It can be mouse, it can be rabbit, some other animal model. So if I say I want to raise antibody against protein A, so this is my protein A. So what I need to do, I have to inject this protein to some, say, mouse. For example, I am saying. So now there is some kind of details protocol how to do this, and after some time you can get some antibodies against this protein A from this mouse blood. Okay, so antibody against protein A. But the thing is, these antibody, they are not monoclonal antibody, because when you are actually like injecting protein A into this mouse, like mouse immune system will work on that, and this protein A might be very big protein, and several kind of peptide antigen can be presented, and as a result of that, it will be kind of mixed population of antibody. So as a result of that, we will not say that this is monoclonal antibody. Monoclonal means it's kind of same, exactly same antibody what we are getting, and so as a result of that, their specificity will be exactly same. So that is called monoclonal antibody. And also engineered antibody I will discuss. Monoclonal antibody can also be engineered, and I will explain soon. And immune checkpoint inhibitor, this is again another very important strategies that we have a different checkpoint when our immune system is getting activated. So we can target those checkpoints also to modulate our immune response. Here, particularly, I would like to focus on monoclonal engineered antibodies to decrease immunogenicity but maintain their antigenic specificity. So now, monoclonal antibody I already mentioned, right? So for example, if you see, this is fully mouse antibody. That means that antibody raised in mouse, like. So it can be, the antibody can be against some human protein, but the antibody we are saying fully mouse means it is completely raised in mouse, and as a result of that, I am saying that fully mouse antibody. And there are some convention that if it is mouse antibody, the antibody name end with this kind of this letter, and if it is chimeric, I will explain what is chimeric, and it will be end up with this letters, and similarly humanized with this letter. That is not that relevant in our discussion, but some kind of convention in nomenclature. Now I will discuss, I will give example of each of these. Now the thing is, why we are trying to reducing the immunogenicity? Because when we are getting the antibody from mouse, then if this antibody is injected to human, it might show some immunogenic reaction against that antibody, because it is after all a foreign protein. So that's why we are trying to make as low as their immunogenicity we are trying to reduce. Okay, so the first one is mouse antibody, fully mouse. So one example is Mudomomab. So as I mentioned that this part you can see this is same like this. But anyway, so what it does, so this is anti-CD3 antibody. It's a monoclonal antibody but anti-CD3 antibody. So CD3 is a marker of T cell. So it is present on the surface of T cell. So as a result of that, this antibody inhibit the T cell activation. So if it inhibits T cell activation, then I would say it will dampen the immune response. Somehow it will be little bit immune suppressive. So that's why this kind of antibody can be used during organ transplantation, particularly before it was used for kidney transplantation, so that during transplantation organ rejection will not happen if the immune system is little bit in its suppressive condition. Okay. And the next one, chimeric antibody. If you see here, we change some portion of this antibody by completely human sequence. For example, this whole Fc region, fragment crystallizable region, and also the constant region here of this variable, heavy chain as well as light chain. All these portions are from human sequence. So only the variable domains of heavy chain and light chain, they are containing mouse sequence. And as a result of that, it will show less, I would say the immunogenicity will be decreased. So it will be much better reagent compared to the fully mouse antibody. So now, if I give you one example, this is rituximab. So again, if you see that this is matching here, okay. And one example, like already I mentioned, so this is against anti-CD20, and it eliminates B cells, and this was used in non-Hodgkin's lymphoma to treat this kind of disease. This antibody was used, and then like scientists, they are trying to make it even better. So that is called humanized monoclonal antibody, where you can see everything replaced by human sequence, only that CDR region, the complementarity determining region, complementarity determining region. I told you CDR 1, 2 and 3 here. So just keeping those things, then like other portions are human sequence. So one example here, this one. So this is against anti-IgE. So Ig is related to some allergic reaction. Also, it has some some kind of correlation, and this antibody removes IgE antibody, and as a result of that, this is used against chronic asthma. And the last one, this is very important, that fully human antibody, monoclonal antibody. One example is this is ipilimumab. It's kind of tongue twister, right? So ipilimumab. This is against CTLA4. Like CTLA4 is some kind of receptor, inhibitory receptor present on T cell. I will be discussing a little bit in detail soon. And it increases the T cell response, and it is being used now against metastatic melanoma. So this is some kind of approach, some kind of monoclonal antibodies, and particularly engineered antibodies for the therapeutic purpose, like either we used before or currently in use. Now, I would like to discuss a little bit more engineered version of antibody, and one of that example is bispecific antibody. So then what is bispecific antibody? I mentioned before that in case of antibody, like if I say this is one heavy chain and the next heavy chain, two heavy chains, right? And two light chains. This is some kind of antibody. And here, particularly I mentioned that it will capture one antigen here. Similarly, in this region, one antigen. But for an antibody, this antigen will be same antigen, right? Because these two arms, they are identical. Same antigen should be there. Now, the bispecific antibody means they will, they will recognize two different antigens. This is some kind of engineered antibody. Let's see how we can make that. So, in order to make that, so I'm just giving you one example. Catumaxomab, this is some kind of bispecific antibody, and it was used particularly in some European country against some cancer. Okay, so I would like to discuss what is happening here. So this antibody is made up of in this way. So if you see, as I already mentioned about the antibody, so this is one antibody, and this antibody recognize some antigen which is coming from EPCAM. Full form is epithelial cell adhesion molecule. Okay, so this is some kind of protein, and this antibody is against that. So now, if you see another antibody I am talking about here, so this antibody is against CD3, So CD3 is, as I already mentioned, it is present on T cell. So it recognizes CD3. So now we designed a single antibody where one arm is something like this, that is holding EPCAM, and the other half is coming from here. So one half from here and the other half from here. So as a result of that, this one I am just changing the shape of this antigen so that it will be easy to understand. So this is the case here. So as a result of that, this is coming from here. Okay. So this is holding, this is against CD3. So this antibody, I would say this is bispecific antibody. So what are the applications of bispecific antibody? So it will be very useful in such a way, like, I would like to explain that. For example, if you use this antibody, so first I would like to mention here, if you see this left part and right part is different here. This is against CD3 and this part is against this epithelial cell adhesion molecule. So now if you apply this antibody, what will happen? So I would say, if you have this is cancer cell. Why? Because this epithelial cell adhesion molecule, they generally get overexpressed, they express in some cancer condition, not in normal cell. So we have to design in that way. So this is present in some cancer cell. So I would say, if this is the EpCAM protein, so epithelial cell adhesion molecule here. Similarly, so what will happen? The antibody will bind here, and the same antibody, since it's bispecific antibody, here it will bind to CD3, and CD3 is present where? It is present in T cell. So therefore, this antibody is driving the T cell to come close by of this cancer cell. So these two cells are very close to each other. Then what will happen? T cell will exert its effect, so it will try to kill the cancer cell. So this is the strategy. On top of that, what is going to happen? As you know that in Fc region, so Fc region will bind to some Fc receptor. So as a result of that, so if this is Fc receptor, it can be, I would say, NK cell, or it could be macrophage. So now I hope that you understand what I am trying to say, that this antibody is driving this T cell and NK cell or macrophage very close by of this cancer cell, and therefore it can exert its effect. Okay. So this is how bispecific antibody work. And now I will explain another example here. This is called antibody drug conjugate. So there are many antibody drug conjugates are already in use as a therapeutic reagent, and here particularly I am mentioning this one, enfortumab vedotin. So this is again a monoclonal antibody against this protein Nectin-4. So Nectin-4. So the antibody is against Nectin-4. So Nectin-4, this protein, this is generally present during embryonic stage, not in healthy adult, in human I am talking about. In healthy adult, the expression of Nectin-4 is very, very low. In most tissues, they don't even express Nectin-4 in healthy adult. But in cancer condition, Nectin-4, expression of Nectin-4 increases in many different types of cancer, right? For example, renal cancer, different types of lung cancer, this Nectin-4 expression again reappear. So as a result of that, what scientists did, they developed some kind of monoclonal antibody against Nectin-4. So I would say, if this is the monoclonal antibody against Nectin-4, now this antibody is again attached with some drug, and the name of this drug is, this is some kind of drug, and here the name of this drug is, this is some kind of chemotherapeutic agent. So particularly this is monomethyl auristatin. So this is MMAE in short form. Now, what is its function? So the thing is, then this antibody will bind to Nectin-4, which is like being expressed by some cancer cell. Now some of this antibody will go inside the this cell, because cell will endocytose some of this Nectin-4 antibody. And now I already mentioned this antibody is attached to this drug. So now this drug will exert its effect. So this drug, particularly MMAE, so it disrupts microtubule, okay, disrupts microtubules present in the cell, particularly cancer cell, because it is only going into cancer cell, because cancer cell is expressing Nectin-4, okay. So now as a result of that, it will induce cell death. So this is some kind of strategy of like conjugating drug with a specific antibody to target cancer cell. And this is FDA approved for the treatment of locally advanced or metastatic urothelial carcinoma, bladder cancer, okay. And now I will discuss like about immune checkpoint inhibitor. This is the last discussion of this immunotherapeutic part. So now here if you see, like I particularly mention antigen presenting cell APC, they present antigen through MHC molecule. So this is our antigen, peptide antigen. And TCR or T cell receptor, they recognize the peptide loaded MHC molecule, and then the T cell activation and all those downstream things happen. So I just mentioned that. I didn't discuss in detail what are the other things that are happening, but now I would like to mention, otherwise I cannot explain this. The thing is, this interaction, this is, I would say, very like, if I give you some kind of analogy, this is some kind of, I would say, if you are trying to drive a car, so you need a specific key. So this key required for the ignition of that car. So this peptide MHC and TCR interaction, this is so specific. This is just like a particular key is required for that particular car. Now, after the ignition, you are just sitting there inside the car. Then what will happen? The car will not move. So in order to moving the car, so we have to press the gas, the accelerator, right? So as a result of that, car will move on. So similarly here, just this recognition is not enough. You need some additional signaling mechanism. So the first thing is, you have receptor here. This is called CD28, very important receptor present on T cell, right? Particularly those are like T cell and naive T cell. They are just saying antigen now. So CD28, it interacts with some ligand. It's called B7-1 and B7-2. So B7-1 and B7-2 present on antigen presenting cell, and CD28 is present on T cell. When it interacts, then it will provide some kind of signal, which is called stimulatory signal. So just like I mentioned the analogy that you are putting pressure on accelerator so that the car will move, so that T cell will function now. Now, if you just moving your car going on and on, but sometime after seeing some stop signal or something, you have to stop also, right? So you have to put the brake. So in T cell also that kind of mechanism there, that is kind of homeostasis is required, because if our immune system is too active, that is not also good. So we need some kind of optimal function of T cell. So it cannot be like that it is always giving the stimulatory signal, always it is activated state. That is not good. So as a result of that, at the beginning the stimulation is required, and then after some time, eventually another receptor will be expressed on this T cell. So this is called CTLA4. It was not there at the beginning, but after some level of activation, CTLA4 get expressed, and as a result of that, it will also bind to the same ligand B7-1 and B7-2, just like CD28 interact with B7-1 and B7-2. But this interaction, CTLA4 and B7 interaction, is much stronger, almost I would say 10-fold, 20-folds higher in terms of affinity in compared to CD28 and B7. So as a result of that, although CD28 and CTLA4 they are competing for the same ligand, but since the affinity is more with CTLA4, it will bind to B7 molecules, and then it will provide you inhibitory signal to that T cell, so that the T cell homeostasis will be maintained. So it will not be overactivated. So this is some kind of balance, and this is called T cell co-stimulation, and this is very important for proper immune function. Now, after understanding that, like in biomedical field, scientists they develop some therapeutic reagent, particularly different types of antibodies and protein-based reagent to tackle this system, to modulate this kind of co-stimulation like to tackle different types of diseases. I am giving you just one example. For example, this is FDA approved drug I mentioned during the discussion of monoclonal antibody. This is ipilimumab. So this is also called Yervoy. This is the commercial name produced by Bristol Myers, and this is currently being used against melanoma. Now what is this? So this is an antibody, monoclonal antibody, that binds to CTLA4. So it binds to CTLA4, and then it blocks this inhibitory pathway. So then why we are using this? That means during different types of cancer, particularly in melanoma, what we need? We need more activation of T cell so that they can clear the infection. It's not infection, the cancer cell. So as a result of that, we are trying to prevent this inhibitory pathways. So we want that T cell remain more and more active. So it is, as I mentioned, it is already in use. So there are many other examples, but now I will explain little bit some other reagent which can be immunosuppressive. Here, another FDA approved drug, it is called Orencia. What is this? This is soluble analog, soluble form of CTLA4 with Fc fusion. Fc means that part of constant domain of that antibody, right? So that Fc region, I already discussed about that. So CTLA4 fused with Fc. Now, what will happen? This can be used as immunosuppressant, and it can be used against some autoimmune disease, for example, rheumatoid arthritis. How? Because this CTLA4 is not membrane bound, so it is soluble CTLA4 I am talking about. So now if you are adding CTLA4, what will happen? This CTLA4 will bind to B7, right? B7-1 and B7-2, because I already mentioned the affinity between CTLA4 and B7 molecules are much higher compared to CD28. So as a result of that, if you administer this soluble form of CTLA4 fused with Fc, which is much more stable and better reagent, that's why we are adding Fc region with that particular soluble CTLA4. Now they will bind to B7 molecules, but they are not, this CTLA4, they are not membrane bound, they are not on the T cell, so they cannot provide the inhibitory signal to T cell. Then what will happen? So as a result of that, the CD28 here, they cannot be engaged with the B7 because B7s are already saturated by this CTLA4. So therefore, our immune system, like the T cell, will not be that much active because the CD20, they are not getting B7 on the antigen presenting cell. Because of that, this kind of strategy can be used against autoimmune disease. For example, as I already mentioned, that Orencia, it is being used against rheumatoid arthritis. But there are some pros and cons about this immune therapy also. So we have to critically evaluate, those things, and accordingly, we need to use all these kind of immunotherapeutic reagent. So this is just I just started a little bit of this kind of discussion, but if you are interested, you can follow any good textbook and recent literature. You will get huge information, because particularly immunotherapeutic reagent for recent cancer therapy, it is coming up. Okay, so that's all, and thank you very much.